Just documenting the first small genome was enough to suggest the awesome size of the awaiting larger ones. If all the A's, T's, G's, and C's of the Haemophilus genome were printed on paper of this size, the resulting book would run some 4,000 pages. 2 pages on average would be needed for each of its 1,727 genes. Of these, only 55% have readily identifiable functions. For example, energy production involves at least 112 genes, and DNA replication, repair, and recombination requires a minimum of 87. We can tell from their sequences that the remaining 45% are functioning genes, but we simply can't, at this stage, be sure what it is they do. By bacterial standards, the Haemophilus genome is pretty small. The size of a bacterial genome is related to the diversity of environments a particular species is likely to encounter. A species that leads a dull life in a single uniform setting, say the gut of another creature, can well get by with a relatively small genome. One that hopes to see the world, however, and is apt to encounter more varied conditions, must be equipped to respond. And flexibility of response usually depends on having alternative sets of genes, each tailored to particular conditions and ready at all times to be switched on. Pseudomonas aeruginosa, a bacterium that can cause infections in humans and poses a particular danger for cystic fibrosis (CF) patients, lives in many different environments. We saw in Chapter 5 how a genetically doctored form of a related species became the first living organism to be patented. In that case, it was adapted to life in an oil slick, an environment notably different from the human lung. The Pseudomonas aeruginosa genome contains 6.4 million base pairs and 5,570 genes. About 7% of those genes encode transcription factors, proteins that switch genes on or off. A respectable proportion of its entire genetic complement is thus devoted to regulation. The E. coli repressor, whose existence was predicted by Jacques Monod and François Jacob in the early 60s, see Chapter 3, is just such a transcription factor. A rule of thumb then would go as follows. The greater the range of environments potentially encountered by a bacterial species, the larger its genome, and the greater the proportion of that genome dedicated to gene switching. Tiger did not stop at Haemophilus. In 1995, collaborating with Clyde Hutchison at the University of North Carolina, the Institute sequenced the genome of Mycoplasma genitalium as part of what has been dubbed the Minimal Genome Project. M. genitalium, which despite its ominous name is a benign inhabitant of human plumbing, has the smallest known non-viral genome, some 580,000 base pairs. Viruses have smaller genomes, but by co-opting the genomes of their hosts, can get away with not having the genetic wherewithal for many fundamental processes. And that relatively short sequence was found to comprise 517 genes. So a question naturally arose. Is that the minimal gene complement necessary to sustain life? Subsequent research has set about knocking out M. genitalium's genes to see which are absolutely vital and which are not. Currently, it appears that the minimal genome contains no more than 350 genes, and possibly as few as 260. Admittedly, this is a somewhat artificially defined minimum, since the enfeebled bugs are supplied through their growth medium with every substance they could conceivably need. It's a bit like claiming kidneys are not necessary for life because patients can survive on dialysis machines. Will we ever be able to construct a functioning minimal cell from scratch by artificially combining its separate purified components? Considering there are more than 100 Mycoplasma genitalium proteins whose functions remain a mystery, the achievement of such a goal seems for now a long way off. Even the 500 proteins of mycoplasma, some represented in the cell by a huge number of molecules, some by just a handful, constitute an enormously complex living system. I, for one, have enough difficulty following a movie like Gosford Park, in which there are more than 4 or 5 major characters. The thought of blocking out the complexity of interactions among the vital players inside a living cell is nothing short of mind-blowing. For the living cell is no neat miniature machine. It is rather, as Sidney Brenner put it, a snake pit of writhing molecules. Still, Craig Venter is confident that the era of the artificial cell is just around the corner, and he has wasted no time in assembling a panel of bioethicists to counsel him on whether to venture forth. They, like me, see no moral dilemma in trying to create life in this way. If such a feat were ever achieved, it would merely reaffirm what most of us in molecular biology have long known to be the truth. The essence of life is complicated chemistry and nothing more. Such a finding would have made headlines a century ago. Today, it's no big deal. Only the opposite conclusion, that there is more to the life of the cell than the sum of its basic components and processes, could generate deep excitement in today's scientific world. DNA analysis has already changed the face of microbiology. Before DNA techniques were broadly applied, methods of identifying bacterial species were extremely limited in their powers of resolution. You could note the form of colonies growing in a petri dish, view the shape of individual cells through the microscope, or use such relatively crude biochemical assays as the Gram test, by which species can be sorted out as either negative or positive, depending on features of their cell wall. With DNA sequencing, microbiologists suddenly had an identification factor that was discernibly, definitively different in every species. Even species like those inhabiting the ocean depths that cannot be cultured in a laboratory because of the difficulty of mimicking their natural growing conditions are amenable to DNA analysis, providing a sample can be collected from the deep. Now led by Claire Fraser, Tiger remains the leader of the bacterial genomics pack. In short order, they have polished off the genomes of more than 20 different bacteria, including that of an ulcer-causing Helicobacter, a cholera-causing Vibrio, a meningitis-causing Neisseria, and a respiratory disease-inducing Chlamydia. Their biggest competitor is a group at the Sanger Center. The British contingent is led by Bart Burrell, who had the luck not to be in the United States, where his limited academic credentials would have barred him from Top Gun status. He has no Ph.D., having come into science straight out of high school to work as Fred Sanger's assistant, long before DNA sequencing became a reality. Before moving on to bacteria, Burrell made his name as an automation pioneer, having used several ABI sequencing machines to crank out some 40% of the baker's yeast genome of 14 million bases, while the largely European yeast sequencing consortium remained wedded to manual methods. Burrell's group later had the satisfaction of being the first to complete the sequence of Mycobacterium tuberculosis, the agent of the fearsome affliction once known